Thank you all for joining our webinar. The technologies and methods that our esteemed panelists will discuss with you today are critical for the quality, reproducibility, and therapeutic relevance of assays. Understanding biology is the foundation for creating useful protocols, and of course useful protocols let us better understand the biology. With the Assay Guidance Manual from Nathan, we will look at an important resource for researchers offering best practices and guidelines, and then Professor Roepe will guide us through interesting examples for discovering antimalarials.
GoToWebinar offers us a questions panel where you can pose your questions for our panelists that we will get to at the end of our discussions. Please pose a question at any time. Again, we are joined by Nathan Coussens of NCATS and Paul Roepe of the Center for Infectious Diseases at Georgetown University. Thanks for joining us. Let me launch our discussion by going to Nathan. Nathan, tell us about your research background and experiences at NCI and NCATS and elsewhere.
Certainly, and I want to thank the CDD and the organizers for inviting me to join all of you today. It's a real pleasure. So as an undergraduate, I studied biochemistry at the University of Iowa, and pretty early on became involved in research laboratories that were really located throughout the campus. And actually my first experience involved endocrine research, like steroid research in crabs, in the laboratory of Dr. Eugene Spaziani in the Department of Biology. And during that time, I was introduced to one of his colleagues, Dr. Barbara Stay, who's an insect biologist. And her and I have actually continued to collaborate up to this day, and actually have published several papers over the last 2 years, including one this past summer. And I spent a year in the psychology department assisting Dr. John Freeman with the experiments relevant to neurobiology of learning and memory. And through my coursework, really became very interested in protein structure and function. And for my undergraduate honors thesis, I ended up joining the laboratory of Dr. Ramaswamy Subramanian, who's in the Department of Biochemistry. And Rams really has a diverse research interest, and his lab uses a variety of biophysical approaches to really understand protein structure and function. And the wonderful experience that I had while working in his lab is what I think really made me want to pursue a career in research. And I ultimately ended up doing my graduate studies in Rams's lab. And they were basically focused on studies, mechanistic studies of the innate immune system and host pathogen interactions. And then while I waited for my wife to finish school, I spent an additional year at Iowa as a postdoctoral fellow in the interdisciplinary immunology program, and collaborated with Dr. Michael Apicilla's group in the Department of Microbiology. And we were developing small molecule inhibitors of a host evasion mechanism in pathogenic bacteria, which his lab had discovered. And but really, you know, for the duration of my postdoc, I really wanted to expand my skill set and investigate protein structure and function on a larger scale. And for example, signaling in T cells within the immune system involves these multi-protein complexes, which can be quite large, but below the scale of organelles. And these structures are really critical for proper biological function, but frankly, they're extremely challenging to study because these structures can be really highly dynamic, both in terms of their composition as well as their spatial and temporal assembly. And so I carried out the majority of my postdoctoral studies investigating signaling complexes within T cells in the laboratory of Dr. Larry Samuelson at the National Cancer Institute, who's one of the world's experts in T cell signaling. And it was a really incredible experience and research environment. And his lab is well known for using cutting-edge technologies and really taking diverse approaches to the study of signaling in T cells. And ultimately, you know, looking forward, I definitely was interested in a research career within translational research, and I was thinking the pharmaceutical industry. And towards the end of my postdoc training, really doing a lot to investigate career opportunities at that time. And one of the things that I did was participate in this really tremendous program offered by the NIH, and it's called the Translational Science Training Program. You can look that up online. It's described as this multi-day boot camp, and it beautifully covers the entire bench-to-bedside process. And actually, during this program, we were able to tour NCATS before it became an official center of the NIH. And I felt strongly that the mission of the NCATS is incredibly important, and pursued employment at NCATS and was later hired on as a staff scientist. And at NCATS, I really have been able to draw, I would say, heavily from my diverse scientific training in order to develop biologically and therapeutically relevant biochemical and cell-based assays that are really applied to study a variety of different human diseases. Now, many of my colleagues that work with me at NCATS actually came here from the pharmaceutical industry, and when I joined, I felt that I was missing some critical experience, kind of not having that background. But while I was still at the National Cancer Institute, I had identified the AssetGuidance Manual online and was really excited that Eli Lilly and Company had released this important internal training document to the public. And after joining NCATS, I really made it a point to read that book cover to cover, which at the time was about 850 pages. And I would say that having that opportunity to learn some of the tribal knowledge from pharma through the chapters of the AssetGuidance Manual really meant a lot to me personally. And I was delighted when the editor-in-chief, Dr. Sitta Sithampalam, who actually initiated that manual and really drove the development of it while working at Eli Lilly and Company back in the 1990s, he asked me to join him as an editor. And as my primary role at NCATS is a research scientist, I collaborate with a number of scientists that do basic research and are really establishing a translational focus. And I would say that having come from that environment so recently, I definitely understand some of the challenges that they face. And so as an editor of the AssetGuidance Manual, I try to do as much as I can to help make that critical information available to the researchers who need it.
Awesome. Thanks, Nathan. Paul, maybe would you please highlight some of your malaria research?
Sure. Before I do, though, I'd also like to thank CDD and the organizers, and I hope this webinar is helpful to all of our colleagues out there working to perfect their assays of choice. So my group has been studying malaria for a little bit over 20 years now. Our primary interests have been in elucidating drug resistance mechanisms and then trying to use this information to then assist development of what many of us call second-tier therapy, meaning drug therapy that is effective not only against malaria but against drug-resistant strains of malaria. So we've also studied drug-resistant tumors and bacteria as well as parasites, but the majority of my group these days is focused on questions related to malaria and the parasitic infectious diseases. We've always been a highly interdisciplinary group with students and postdocs in the lab that are studying essentially chemistry or molecular biology, biophysics, and some other disciplines. And then they're coming in and out of the group on a regular basis as they finish their degrees and go on to other things and so on. So that's always been really important for us because anti-malaria drug resistance is a topic that demands interdisciplinary perspective, and we're lucky that we're always learning new techniques and approaches. We're also able to rely heavily on a number of generous collaborators to teach us what we need to know when we need to know it, and so all this makes our lab a fairly vibrant and intense place to work. One of our first projects in antimalarial drug resistance, sticking to this theme of interdisciplinarity and working with collaborators, was back in the 90s and started with collaborative work we did with the Tom Willems lab at the NIH on identification of what's called the PF-CRT gene. Back in those days, Tom's group was in the LPD, or the Laboratory of Parasitic Diseases, in the old Building 4 on the Bethesda campus. And this was 97, 98, as I recall. And Tom's lab was filled with a crop of terrifically smart postdocs and research associates. My laboratory still collaborates with several folks from that old group. David Fiddock and Jinswan Su are co-investigators on 2 of our current grants, and Michael Furvik has published several papers with us in recent years. Back then, they were really very exciting days. Malaria research in this country just before that really was not terribly prominent, and around that time, the mid to late 90s, was starting to gain considerable additional momentum as the problems became a bit more tractable, and the NIH and other agencies began to pay more attention to the disease. So Tom had done a tremendously important experiment back in the early 1990s when he crossed chloroquine-sensitive and chloroquine-resistant malarial parasites and was able to clone out 36 progeny from that cross. About half of them were found to be chloroquine-sensitive and half of them CQR, or chloroquine-resistant, suggesting that the resistance to chloroquine and perhaps other quinoline antimalarials was a Mendelian trait, or controlled by inheritance of a single gene. And as long as resistance is defined by an IC50 shift versus chloroquine, this turns out to be the case, as a paper that we were privileged to co-author with Tom and his group back in 2000 proved. The gene that was described in that paper, PFCRT, remains an active area of research in my group to this day. So the encoded PFCRT protein turns out to be a vacuolar membrane protein that is intimately involved in vacuolar physiology. When PFCRT is mutated in certain ways, resulting in patterns of amino acid substitutions, the protein is able to mediate what we call electrochemically downhill transport of charged chloroquine drug from inside the vacuole. This is also called the digestive vacuole, or DV, to the cytosol of the parasite. And this is a key aspect of the chloroquine resistance mechanism. And when the PFCRT gene was fully sequenced from different isolates that were available back in the late 90s, we only knew of 2 different resistance-conferring alleles. One was found in Southeast Asian parasites and one in African parasites. We named these DD2, and the other one was named 7G8. These were just names that came out of the isolates that came from those 2 continents. But since then, dozens and dozens of additional mutant PFCRTs have been discovered in additional isolates from around the globe. The widely accepted hypothesis is that these reflect different drug selection histories or variable drug selection pressures applied to geographic regions. So I guess all of this underscores the important principle that malaria is a global disease with a high degree of regionally specific variability, and various drugs have variable effectiveness in different regions of the globe. So this highlights our first truly big challenge in assay development for antimalarial drug discovery, because we no longer really screen for drugs versus malaria. We screen versus many different drug-resistant strains that represent malaria, pockets of malaria around the globe. And as we learn more and more about the different subtypes or strains of P. falciparum malaria that are carrying different PFCRT alleles, as well as some other mutations, we and a number of other groups have been better able to more accurately model various drug-resistant phenotypes and therefore screen for drugs and drug combinations that show increased potency specifically versus those subtypes.
Well, can you tell us a bit more about your collaborative work, given all your various roles at Georgetown, the NIH, Notre Dame, Case Western?
They're free-swimming, eukaryotic, single-cell eukaryotic organisms that are parasitic. They infect both mosquitoes and a vertebrate host. There's about 160 species of Plasmodia on the planet, and they're known to infect virtually all known vertebrates. Somebody once told me that even whales can get malaria, but I've never met a whale that has malaria. Humans can be infected by 5 different species of Plasmodia, with Plasmodium falciparum and Plasmodium vivax being the most medically important, the most common. Most deaths from malaria come from falciparum, but there are about equal numbers of vivax and falciparum infections around the world. If you look at the globe, different regions of the globe are biased towards one versus the other. So in Africa, most malaria is falciparum malaria, whereas in India and Pakistan, you might find 50-50. Once within humans, after the bite of a female Anopheles mosquito, these malarial parasites quickly invade the liver. They burrow inside of liver cells, or also called hepatocytes. They develop there asexually rather rapidly. In about a week to 2 weeks, burst out of the liver in these giant clumps of a newly differentiated form that we call merozoites. And the big clumps of merozoites we call merosomes, and they can be as many as 100,000 merozoites in a merosome. So these things hit the bloodstream, break up into individual merozoites that then infect red blood cells. They exist briefly as free blood forms before all of this, called sporozoites. That's the form that's released during the mosquito blood meal. And as I just mentioned, as merozoites released from the liver. So the first key point, I guess, in terms of the insights that requires detailed attention for assays is that the microbe you're really screening against exists in a multitude of very different, highly differentiated forms. If you want to screen against the red blood cell stages, your culture system is going to have to include fresh red blood cells and matched human serum. But if you want to screen versus the free blood form known as a gametocyte, you'll need separate, highly specialized culturing systems to enrich for that form, and so on and so on. So these different culturing systems make screening for compounds effective against most or all of the human stages quite difficult. Drugs active against all forms cannot be found via just...
Okay, so we're going to take a second to actually run a poll. Charlie, if you could put that poll up, we're going to take a few seconds to ask the audience their answer to the following question.
All right, everyone. All of the attendees should now see a poll. So we're very interested from our audience today. What do you see as the major source of variability in your assay? You guys can all select your answer here, whether it's reagent consistency, assay implementation, biological variability, technical or instrumentation issues, or data analysis. Please go ahead and make your selections, and I will close the poll in 5, 4, 3, 2, 1. Thank you very much. So now we can see from the audience a nice 58% majority feel that biological variability is the major source of assay variability. Great.
Very nice. Right. Thanks, Charlie. Okay.
Can I comment on that real quickly?
Yes,
I'm actually quite happy to see that, because that's pretty much how everybody in my lab feels too.
Yeah.
Fantastic.
Yeah, it was also interesting that was C, and it was almost like a perfect Gaussian distribution, with the first and last choice being the lowest.
Very good. Okay, let's continue on. If it's okay, I'd like to ask Nathan more about the Assay Guidance Manual. You included a chapter in there entitled Development and Application of the Bioassay Ontology to describe and categorize high-throughput assays from Vempati and Schur. Could you elaborate a little bit on that?
Yeah, certainly. Actually, I think to address that, I'd like to take a step back and, as I mentioned, the major focus of the Assay Guidance Manual is really on the methodologies and best practices to support assay development and high throughput screening and lead discovery. And the gift of the AssetGuidance Manual from Eli Lilly and Company between 2004 and 2005 was really intended to benefit preclinical drug discovery initiatives worldwide. But this time period was also the beginning of the NIH Molecular Libraries program, which of course involved a number of screening centers located throughout the United States. And this really increased the public access to small molecule science through high throughput screening and was intended to advance translational research. Now, prior to the Molecular Libraries program, access to high throughput screening had primarily been limited to the pharmaceutical industry. And the AssetGuidance Manual was made available to the public from the NIH Chemical Genomics Center website, both to guide the work being done at these different screening centers, but also to really provide the public and increase public understanding of, the resulting data and, the workflow that went into generating the data. Now, the PubChem database was created in order to allow the public access to these very large data sets that were being generated at the different screening centers of the Molecular Libraries program. And it was really the first time that the public had access to this type of data, which included data from primary screens and confirmation screens and secondary assays, such as counter and orthogonal assays. And at that time, there was no easy way for the public to really organize these diverse data sets that would allow systematic querying. So the Bioassay ontology was developed to support public access and use of the chemical biology data from PubChem. And this particular chapter that you asked me about was published in the AssetGuidance Manual so that readers can understand what the ontology terminology is and how that can be applied to data from high throughput screening and used to examine more deeply the information within the database.
Great. I have a follow-up to that, actually, Nathan. But before I ask you the question, I want to remind people that they can pose their questions to the panelists in the questions panel in the GoToWebinar. So please do that at any time. So Nathan, the follow-up was, what should experimental researchers know about emerging informatics technologies to do those jobs better?
Okay. Well, I think that the power of applying informatics approaches to research questions is rapidly increasing, as are the experimental technologies that are increasingly generating large data sets. And together, this is really driving our overall understanding of biology and increasing that. And I think that researchers worldwide are definitely enjoying a growing wealth of data that's comprising very diverse biological measurements. And publicly available informatics tools, including the Bioassay ontology that you mentioned, and of course the CDD Bioassay Express, really allow researchers to utilize these large data sets to develop and test new experimental hypotheses. And additionally, these tools can be used to facilitate multidisciplinary collaborations among, different types of researchers, such as biologists and chemists and engineers and bioinformaticians.
Great, great. And we have one more poll we want to ask. I think it's always interesting to hear what people are thinking. So we're gonna tease you one more time with another poll. Charlie?
All right, so hello again, audience. Please respond here to our quick poll and let us know what aspect of assay development do you currently find most challenging. Choose between physiological therapeutic relevance, technical capabilities to measure parameters of interest, most relevant controls, assay validation, or data analysis. We'll see if we can get a nice little curve here around the middle option again to make our scientific brains happy here. All right, everyone, thank you. I will close the poll in 3, 2, 1, close. So the results here did not, unfortunately, follow the nice curve that we had before. So option A got a majority, 43%, of most people finding physiological and therapeutic relevance issues the most challenging.
That is, in fact, the subject of today's talk, and all these people joining us are clearly interested in therapeutic relevance of bioassays. So I think that makes sense.
I do. I like it when it happens like that. Thank you all very much.
Okay, continuing on to Paul. Paul, both target and phenotypic-based approaches to drug discovery offer advantages and disadvantages. How do you view and apply these different approaches in your own research?
Yeah, sure. So as Nathan was just mentioning before the poll, we almost we almost have too much data out there that's hinting at more and more targets, and targets that intellectually, scientifically seem quite attractive, but as the poll just illuminated, we often don't know how physiologically relevant those targets might be with respect to controlling the disease. So I think our group, like a lot of groups out there, hedges our bets, and we basically pursue both. Many times, depends on the drug class or the drug lead, but many times there isn't really well-formed or preformed notion as to which type of screening is going to be best for us. Sometimes it's just what we have available at the time, and we're just doing it the best that we can. But I suspect as our understanding of malarial parasite biology and the proteome in particular continues to improve, that we'll see the development of more additional approach. I also, as in a lot of people, the advent of technologies can help us quickly through attractive parts that are omics investigations.
I would like to ask, Paul, I would like to ask you a question.
Sure, go for it, Nate.
Sure.
So I was wondering, when you're thinking about developing screening assays, how do you personally kind of weigh the options and kind of take into account all the different relevant factors, say from epidemiology to drug resistance to malaria biology, to kind of balance that with the assays that you think about developing?
Right. That's a very good question. We agonize over these things quite a bit in my group. For me, I rely quite a bit on my research group to help me stay up to date, to know. A lot of that comes from the literature, but a lot of that also comes from meetings and discussions with colleagues and so on. With a diverse interdisciplinary group, I guess that's a bit easier than you might expect. Each week we have journal clubs and data presentations where individuals in the group take turns presenting what they're interested in, and almost always that presentation will include deep or critical evaluation of literature. And so since the interest across the group vary, as a group level, we manage to stay up to date, pretty well informed. Most of the things that I need to know for the latest round of asset development that we're pursuing in the group. And of course, collaborators outside the group are a huge help in all this as well. In general, the malaria community has a lot of very generous people that are quite collegial about sharing information. So most of what we've been able to perfect in my group in terms of assay development has come through these collaborative interactions with others, and then the homework that the people in my group do.
Paul, you were interested in the public malaria data in CDD Vault, and I'm just kind of curious how relevant and helpful are these malaria data sets, such as the MMV Malaria Box, Johns Hopkins and NCAT FDA-approved libraries, GSK's Trace Contest anti-malarial set. Basically, how can we use our knowledge of previously screened compounds, resistance, and approved drugs to best focus our future anti-malarial drug screening efforts?
Sure. So all of these compound libraries have been extremely helpful. There's a number of drugs in stages of development now that 10 years ago. The field has come a long way in part because of the availability of these compound libraries. So now we have many compounds that can develop better drugs. There's is remarkably better than it was just 10 years ago because of the, again, because of the accessibility of these tools that you mentioned. But what I think needs to happen next is example the work we're doing with Craig Thomas and his group with the NCATs. So what I mean by that is even though most laboratories screen quite capably for antimalarial activity one drug at a time from these fluorides, in the field, in the clinic, we don't use monotherapy anymore. Monotherapy has largely been in favor of drug combinations. There's some powerful logic behind why we want to use drug combinations. The simplified reason is that using 2 drugs at once, if you're picking the right drugs, frequently avoids development of resistance to one compound in a short period of time. So drug combination now become the norm, the standard in treating malaria around the globe. But paradoxically, in the community, we really don't tend to screen for drug combinations. For drug combinations, there are a number of differences of opinion on how you might want to prioritize what comes out of those screens. Obviously, the combination needs to be potent and safe, but my opinion, which is just my opinion, is that it's also best if the drug combination shows potent synergy, higher the synergy. So we're just really at a stage in the field where screening for these sorts of traits, combinations, using these very large collections of possible drugs and drugs leads is really the big new challenge. Can I ask a question?
Yeah.
Can you tell us about the quarter research workshop that you organized?
Yeah, certainly. So after Eli Lilly and Company released the AssetGuidance Manual to the NIH between 2004 and 2005, we wanted to make the information, of course, available to the public, and several different publication platforms were explored. Actually, initially, the entire document could be downloaded from the NIH Chemical Genomics Center website. And then in order to really allow that thing to grow, as it had done within the company infrastructure, we explored this wiki format for a period of time, but ultimately the authors felt that the format was difficult to work with. So in 2012, the content was actually published as an ebook on the National Library of Medicine National Center for Biotechnology Information bookshelf. And as we see it, there are 2 major advantages of the current publication platform. So first, as I mentioned earlier, these ebook chapters have PubMed citations for the contributing authors. And that's, of course, really important for recruiting new authors. But I think it's really critical for the visibility of the content, because much of this content is really geared towards helping researchers. And PubMed is, one of the primary places where these researchers are going to find the information. So it at least exists now where they're going to, be looking for it. Another thing is that the format really allows the ebook to, as I mentioned, exist as this living document that can continue to grow with contributions from researchers worldwide, which was, kind of a long-term vision that we had for the AssetGuidance Manual. And so we actually update this content quarterly to keep, the content current. And as of 2013, we began to be able to track the number of times the AssetGuidance Manual content was accessed. And we've been delighted that over the past 3 years, we've seen that the viewership is doubling from 50,000 retrievals of the content quarterly in 2013 to nearly 100,000 per quarter in 2016. And, as I mentioned, my primary responsibility at NCAT is in collaborations with academic and government researchers to help them develop different types of assays that are suitable for high throughput screening. And I was aware not only from seeing the substantial growing popularity of the AssetGuidance Manual, but also certainly from my interactions and conversations with collaborators, that there's definitely a strong interest among many researchers in learning more about assay development for high throughput screening and late discovery. And when you think about it, an assay really is or a collection of assays are the heart of any drug discovery and development program up until the point where the molecule reaches the clinic. And so the Assay Guidance Manual editors got together and decided to establish a workshop which focused on a variety of critical concepts that originated, and can be found within the manual. And so many of the workshop instructors have 20 to 30 years of experience in the field of drug discovery. And these workshops are designed to allow participants to really take advantage of the instructor's tribal knowledge and their practical experience of how to develop and validate these assays. So up until date, I would say that we've held 5 workshops. The first one was held here at NCATS, and it included tours and even discussions about laboratory automation instrumentation. And then we held 2 workshops at the U.S. Food and Drug Administration, and one of them spanned 2 days. And more recently, on October 27th, we held a workshop at the Promega Corporation following the International Chemical Biology Society Conference on Translational Chemical Biology. And that was in Madison, Wisconsin. And we actually had participants in that workshop from around the world, as far as Australia and Japan. And so last year we held a workshop in association with the annual International 2016 Society for Laboratory Automation and Screening, or SLAS conference. And due to the success of that event, they've actually invited us to continue to hold this workshop in association with their conferences. And so we'll be holding another workshop in February at the 2017 SLAS conference that'll be held in Washington, D.C. on Saturday, February 4th, 2017. And if you're interested in signing up for this, you can see on the screen right now that there's a link that you can go to, and I highly recommend it.
Terrific. That sounds wonderful. I might have to send a few of my people up to the next one. Can you also tell us what topics have been added recently to the Assay Guidance Manual, and what topics you might like to see added next? Maybe there are things that groups out there can help you with.
Certainly, certainly. So we've actually recently introduced entirely new sections to the Assay Guidance Manual, including a new section on assay artifacts and interferences, which already contains 3 chapters written by leaders in the field, and there are several more chapters in development that will be in that section. We've also just added a section on pharmacokinetics and drug metabolism with a new chapter by Dr. T.C. Chung and his colleagues. Within our in vitro cell-based assays section, we recently published a chapter on the application of cellular thermal shift assay, or CETSA, primarily geared towards high-throughput screening, and that was written by Dr. Tomas Lumbach and his colleagues. And for those of you that don't know, the CETSA methodology really provides a powerful and unprecedented approach to validate target engagement within the context of a living cell. I would say another exciting recent addition to the Assay Guidance Manual is a chapter on in vitro 3D spheroids and microtissues by Dr. Jens Kelm and his colleagues, which of course is a rapidly emerging area of interest in assay development. At the moment, in terms of content we'd like to introduce, at the moment we have 26 chapters that are in different stages of development, which really focus on a wide range of topics, all the way from guidelines to assist reviewers of high-throughput screening grants, all the way to medicinal chemistry for biologists. And in terms of topics we'd like to add, we'd really like to see new chapters that describe assay development using stem cells, chapters on phenotypic screening and 3D cell constructs. I'd like to introduce a whole section on biophysical methodologies that can be applied for lead discovery, medicinal chemistry chapters, pharmacokinetics, toxicity and safety studies, drug metabolism, and we want to definitely have a section devoted to biomarker assays. So all these things we'd love to add in. And if any of you listening in today would like to contribute or know of additional content that you would like to see added in, please don't hesitate to contact the editors of the Assay Guidance Manual, and the contact information is available from the website.
Awesome. Nathan, can you walk us through a few highlights from other sections of the detailed assay guidance manual, how they might be helpful for the global drug discovery community in both academia and industry?
Yeah, certainly. So the first chapter of the Assay Guidance Manual actually is written to provide guidelines for both academic researchers and startup companies to assist in planning for different types of drug discovery projects. And it begins with initial target validation and goes all the way up to the point of early stage clinical trials. And the information includes detailed timelines, estimated associated costs, which, by the way, were updated just this last summer for the current, and key decision points when they need to be made, different definitions for vocabulary, and even potential sources of funding. So following that first chapter are 24 chapters on different types of biochemical and cell-based and in vivo assay guidelines. And so, for instance, the in vitro biochemical assay section contains 9 chapters, which cover a variety of important topics. Of course, there are many chapters about enzymes, including a chapter about validation of identity and, how do you validate enzymatic purity. There's a chapter describing the basics of enzymatic assays for high throughput screening, a chapter on, how to perform mechanism of action assays, and there are, chapters for specific types of enzymes. But there are also chapters that cover other types of biochemical assays, such as protein-protein interactions, receptor binding assays, and immunoassay methods. The largest section of the Assay Guidance Manual is the in vitro cell-based assays chapter, and it contains 14 chapters at the moment, including the Assay Guidance Manual's most popular chapter, which is the cell viability assays chapter by Dr. Terry Riss and colleagues. And I'd like to mention actually that chapter was just updated recently to include a section about novel real-time assay that's capable of monitoring live cells for days in culture. And the cool thing is that it can be multiplexed with a variety of other assay chemistries. So if that's of interest to you, I would definitely recommend checking out that chapter. We also have a chapter about cell line authentication by Dr. Yvonne Reed of ATCC and colleagues. We have different chapters about image-based high-content assays, chapters about ion channels, cell-based RNA interference assay development, and cardiomyocyte impedance assays. And all of these chapters provide readers with, suggestions for robust assay methods, but also include tips for assay optimization, troubleshooting, and data analysis. We have a rapidly growing section which is devoted to assay artifacts and interferences, and a section with 5 chapters that are all focused on assay validation, operations, and quality control. And these are all very popular chapters and important and, a major focus of these workshops. We have several sections on assay technology and instrumentation, and these chapters describe some of the specialized equipment utilized for high throughput screening. And that's important even for folks that aren't necessarily doing the high throughput screening, because for example, there are often challenges associated with the adaptation of an assay that might be performing perfectly well in the benchtop, but when it's transferred to a fully automated system for high throughput screening, oftentimes that can be a bottleneck. So successful adaptation really requires an understanding of the instrumentation that's commonly used in high throughput screening, and this is all described in the Assay Guidance Manual. We have a new section on pharmacokinetics and drug metabolism, as I mentioned, that's definitely growing. And the last chapter is a glossary that includes terms found from throughout the book. I would like to ask Paul a question. Paul,
Sure.
You are well known for applying really multidisciplinary approaches to your own research problems. And I know that you've developed a number of assays and methodologies that have advanced anti-malarial drug discovery. And I wanted to ask, are there malaria targets or aspects of malaria that you feel hold promise for therapeutic development, but just remain challenging, either due to technological limitations or a lack of appropriate assays?
brings to mind is we still don't even know what over half of the proteins in the people from, do and unknown. Identified. So identification very, very difficult. And so if you identify an unknown function protein as a target, then obviously developing assays querying that protein can be very, very difficult. So we and many others are struggling with this right now.
Paul?
Looks like Paul's having some difficulties on his end.
Paul, can you hear me?
I'm trying to understand what.
Can I ask another question? You studied, I know throughout your career you've studied drug resistance in both cancer and malaria.
Specific proteins are. Hello?
Hey, Paul, we're having a hard time hearing you.
I can hear you fine. I can hear me. Now I can't hear Nathan.
I'm not speaking.
Yes, we definitely have a technical issue here.
Some type of issue.
Yeah, should we skip to the next question? I think Nathan had something you were going to ask Paul back on that one.
Paul, I was wondering, I know that you've studied drug resistance in both cancer and malaria, and I was wondering if you could highlight some of the similarities and differences that drive resistance in these cells. And also I wondered if, you're investigating drug resistance in these very different types of cells has raised any interesting research questions that you're following up on.
Sure, a couple of them. One simple concept that I think is that's a very far-reaching consequences and that's applicable to both tumor drug resistance and malarial drug resistance is that we now understand that drug resistance can be either intrinsic, meaning that the tumor or the strain of malaria is naturally less susceptible to a given drug prior to drug exposure, or extrinsic, meaning it is either induced or selected for by the drug or some other stress. And some research showed that the mechanisms of these might overlap in certain examples, but more often they're probably distinct. Another shared feature between these two that I think is very, very important, but that has been somewhat neglected in drug screening, is that there are typically at least 2 layers of resistance. And what I mean by that is that most antimalarial and anti-cancer drugs are both cytostatic, meaning that they slow the rate of cell growth, and cytocidal, meaning that they kill the tumor cell or the parasite cell. And often the static and cytal mechanisms of action are different. For example, zinc alkaloid drugs used in the cancer clinic are well known for slowing tumor cell growth by interactions with tubulin, but they often kill by inducing apoptosis. So in these sorts of examples, if the mechanism of drug action has 2 layers to it, then the mechanism of resistance to the drug can also have 2 layers. And so often when we are studying a single example and get an answer for how cytostatic resistance works, sometimes we stop looking, which leaves the problem half unsolved in my mind. But we're currently grappling with that in the antimalarial drug resistance projects.
Wow. Is there time for me to ask Paul another question?
There is. Yeah, let's see how Paul continues to be connected to us.
Okay.
We had some issue there.
Yeah. Okay.
Well, currently, as you know, there's intense activity worldwide to develop more therapeutically relevant assays that incorporate multiple cell types and microtissues. And I know that many researchers are finding that experimental results can be dramatically different when they compare like traditional methods for cell-based assays to some of the more recent 3D systems. And as an example, Dr. Michael Gottesman of the NCI has reported measuring very different expression of genes that contribute to drug resistance in cancer cells when comparing results from monolayer cultures against some of the more advanced 3D models. And I wondered, in your own research, have you had any similar experiences that are maybe prompting you to investigate some different types of cell-based assays?
Oh, sure, sure. And I guess the simple parallel to Mike's work would be the differences that many people see when comparing parasite isolates versus laboratory strains of those parasites that are established in culture in the laboratory. So an isolate, this refers to the parasites directly out of the human body. And if we try to passage them in culture in the laboratory, often their characteristics, including their sensitivity to various drugs, can change over time. Sometimes those changes are slight, but sometimes they're a bit more dramatic. So that's one big challenge. And then the other one, I guess, is animal models, because we tend to, after the in vitro work, we tend to go into an animal model, and the standard in malaria drug discovery these days is something called the Peters suppression test, where malaria parasites are injected into a mouse, and then drug is administered simultaneously or shortly after, and we monitor how increases in parasitemia over time are suppressed by the presence of the drug. But that's actually not how we treat people in the clinic. In a clinic, somebody comes in with a fever and they have a very high parasitemia already present in their bloodstream, and we use a drug not to prevent the expansion of the parasite population, but to rapidly kill several logs of parasites in a few hours. So an infected person might have tens, even hundreds of billions of parasites. We need to get that parasitemia down to below 1 billion in 3 to 6 hours, or that patient's not going to do terribly well. Clinically, we're trying to kill malaria parasites. Often, for an animal model, we're really screening of parasite for mentally two different factors.
While we're dropping off, I wanted to see, Charlie, do we have any questions from the audience?
Yeah, so we actually do, and thank you all very much here for posting a few questions to our panelists, and it's certainly not too late, so if you want to go into your GoToWebinar panel and find the questions section, you can post more questions. We do have one question here that says biological variability is indeed a major contributor to the development of therapeutic relevant assays for drug discovery. How is the AssetGuidance Manual addressing this specific challenge of biological variability? Maybe that's for Nathan.
Yeah, so I think that answers to this can be found peppered throughout the AssetGuidance Manual. And it's definitely a challenge. And this is an important part, I think, of developing assays. You know, and really finding out, it's always a balancing act of identifying assays that are very robust, meaning that they can reliably yield, reproducible results despite small perturbations that can happen or inconsistencies in, the way the experiments perform. For instance, in high-throughput screening, many times you're doing a number of assays at one time, and so really when that happens, it's not uncommon for something to happen unexpected, like maybe the circulation in the room changes, the temperature a bit, and your assays have to be able to withstand that. So you have to balance the challenge of getting a robust, reliable assay with something that has biological relevance. And sometimes when you try to achieve more biological relevance, you do in fact see more biological variability. Now, some of that can be mitigated to some extent by really validating the reagents. And a lot of our chapters do talk about ways to validate the reagents and materials used in an assay. But there are definitely a number of challenges in developing a very, robust, therapeutically relevant assay that, has controlled biological variability, because it really does exist. I mean, definitely biological variability is an issue that affects all of us.
Excellent. Thank you very much. And I think we'll take, we're right at the end time for the webinar, but I think I'll take one more question here from our list of questions posted by our attendees. Do you guys think that the ever-increasing commercial kits that are available have improved the reproducibility or quality of assay data, or has it had the opposite effect? And do you have any evidence either way? So how are these commercial kits affecting the quality and reproducibility of assay data?
I'm happy to comment on that if you'd like. For professionals, meaning people that are highly experienced, that understand molecularly what is going on in an assay, sometimes those kits can be very, very helpful because they obviously speed things up. But I find, particularly when graduate students, postdocs in my group that are relatively new to some of these assays, I actually don't want to use kits because I want them to understand molecularly what's really going on in the assay. And if they don't, often it's very difficult to troubleshoot when things go wrong.
Perfect. Nathan, any follow-up there on your end?
Yeah, I'm sorry. Can you repeat the question one more time?
We were just discussing here the commercial kits and whether or not they are affecting the quality of the assay data.
Yes, so I definitely work all the time with most recent commercial kits, and from my perspective, I'm seeing a lot of really, really outstanding reagents on the market. I mean, really outstanding, and in my opinion, these are definitely having a major improvement on reproducibility and really, fantastic assays. So me and my colleagues, think that this is a good thing, and we're seeing a lot of excellent assays available.
Great. Well, thank you all very much for posting your questions here. We'll do some follow-up through our blog after the webinar here today for any additional questions that were posted through. And we'll send it over to Kellen.
Yeah, I just want to thank the audience again for attending, and we'll be listing some future webinars on our website coming up next in Q1.
Yes, we want to also thank Nathan Coussens and Paul Roepe for their time and preparing for this and really sharing some really good scientific stuff here today. We really appreciate you sharing your experiences with the entire group. So good day, everyone.
All right, everybody. Thank you very much. Have a great day.